Hello everyone, this is Savannah. Today we'll be doing genetics worksheet number 3. Like always, we have our lecture review section, and then we have our practice question section at the bottom. Okay, so for number 1, what stage of meiosis constitutes the law of segregation? So, keyword here is segregation. The phase that we segregate things is anaphase. And this law is referring to genetic variation. So this is going to be anaphase 1. Right, that's when we separate our homologous chromosomes. So the stage the meiosis constitutes the law of segregation is anaphase 1. Now, number 2. What stage of meiosis constitutes the law of independent assortment? So keyword here is assortment. We assort our chromosomes at the metaphase plate. So this is going to be metaphase 1. Right, this is highly testable. I would know this. I would put a star next to this. Okay, so then kind of to piggyback off of that, we're going to ask, what are 3 ways for genetic variation? So the first one is crossing over. Right, that happens during prophase 1. And then we have the law of independent assortment. Which is metaphase 1. And then the law of segregation, which is anaphase 1. I would know this. This is 3 good ways for genetic variation. Okay, so question number 4 asks you to define isogamous and heterogamous. So let's just break down the words, because I think that helps to understand what we're actually looking at. So this prefix right here, iso, this means the same. While hetero, like heterozygous, means different. And this root word, gamous, means gametes. So when we see isogamous, that means across male and female, the morphology of the gametes are the same. And then opposite of that is heterogamous, and that is when they're not the same. And that's what we are, because we have sperms and we have eggs, and those are not the same morphology. They are very different. So that's what those terms mean. So question number 5 is asking to describe spermatogenesis. For this exam, I think you should just have a general overview, be able to compare and contrast spermatogenesis and oogenesis. So for spermatogenesis, we're going to start off with spermatogonial cell. This is our diploid sperm cell. And then this spermatogonial cell is going to divide into a primary spermatocyte. This will then turn into a secondary spermatocyte. And then the end result will be 4 haploid spermatids. And these are unique because they've undergone meiosis. So this is an overview, and then one more thing is this occurs in the testes. So now for oogenesis. It's very similar to spermatogenesis. The end products are not the same though, and it's not as continuous. The males are going to always continue sperm. They'll make thousands and thousands and thousands of sperm, while oogenesis, you're born with a certain number of eggs. They're created in the womb. So it's a bit more pricey, it takes a bit longer, and we'll see that it actually doesn't continue through meiosis until fertilization. So for oogenesis, we're going to start off with our diploid cell, which is our oogonia. This oogonia is going to turn into a primary oocyte. Then, just like spermatogenesis, it'll turn into a secondary oocyte. And then lastly, we're going to have our egg. But because eggs are expensive, we can't make 4. So instead, we make 1 big egg and 3 polar bodies. That's a key difference you should know between spermatogenesis and oogenesis. And then, like we mentioned, this oocyte will be arrested and won't continue meiosis until it's fertilized, because it is a very costly, energy expensive process. And then oogenesis can occur in the ovaries, and then it's not going to finish meiosis until fertilization. So that's kind of the overview that you should know. Alright, so for number 7 and 8, this is dealing with plants. And plants are a little weird because they can skip generations of being haploid and diploid. So our gametophyte is our haploid generation. And then our sporophyte is our diploid. And then the sporophyte will produce spores, which are haploid. And it's kind of like the sperm equivalent for plants. Right? Our spore is like our spermatogonial cell up here. And it's going to produce sperm, or in this case, spores. Okay. So for number 9, this is kind of an important concept. It is, what is self-fertilization? So self-fertilization is whenever this is for plants, mind you, we can't self-fertilize. Self-fertilization is when a plant contains both the anther and the pollen, right? Like this is what Mendel's pea plants would be. And normally, if you're not self-fertilizing, you'd have some kind of bee that comes in, takes this pollen, and then goes to some other anther. So pollen are the human equivalent of sperms, and then this whole anther thing is the pollen has to travel down this anther tube and get fertilized at the egg at the bottom. So self-fertilization is when the plant's anthers and pollen come together and create an identical copy. That's what we see Mendel did a lot with his experiments. In another sense, it's like, let's say we have a heterozygous plant and it's asking you to self-fertilize. So to self-fertilize, you would just cross it with itself. And of course, you'd set up your Punnett square, big P, little p. So that's what it means by self-fertilization. Just breed it with itself. And it's super easy to do in plants, especially pea plants, because you can just keep the flower closed and not touch it, and it'll self-fertilize. So number 10, what is cross-fertilization? This would be our whole bee scenario, right? Our little bee comes in, goes to the pollen of one of the flowers, and then the bee travels to another flower and then fertilizes that flower. So it's plant gets bred with a different plant. That's what cross-fertilization is. Okay, so number 11 is another really important topic, and that is true breeding. So true breeding is when it gets bred by a variety that will always produce the same offspring after several generations of self-fertilization. So an example of this would be our homozygous dominant and recessive. So if we have big A, if we self-fertilize big A with itself, it'll always produce a dominant trait. And if we have a recessive homozygous, if we self-fertilize, we will always get our recessive trait. That's what true breeding is. You also will use this when you solve for an unknown. For the unknown ones, you want to use the recessive trait, the recessive alleles. And we'll see an example of this. But if you're trying to solving unknown, use recessive trait or recessive alleles. If you use dominant, it's not going to tell you anything because it's still going to express dominant even if it's heterozygous, right? Okay, so number 12, explain the P1, the F1, and the F2 generation with true breeding. So this is going to be your ratios that you'll have to memorize. So your P1 is your parent. In Mendel's experiment, we can look at either the monohybrid or a dihybrid. So I'll just break it up into, this one will be mono, this one will be di. So for the parent generation for a monohybrid, he bred big P with little p. And then F1, F just means filial, which is just the kids basically. That would be if we cross big P with little p, they'll always get a heterozygous. And then that'll be self-fertilized by itself. So then we get our F2, which is our ratio. So if we just draw this out real quick. Get our big P, little p, big P, little p. So we get a 1:1 ratio. So this is our big P, our big P, and then our little p. And that's our genotypic ratio. If we're looking at phenotype, then we have these three that express the dominant trait. So our phenotypic ratio is 3:1. So for F2, that's going to be 1:1 for genotype, and then 1:3 or 3:1, which is for a phenotype. Now for the dihybrid cross, we use two different sets of alleles. So for the parent one, he used something like big P, big Y, and we bred it with little p, little y. That's our parent. And then we create all heterozygous in our F1. Right, and then F2 is going to be our ratios. I won't draw that one out until we get to number 15, because I'll show you there. But trust me right now, we'll look at this in a second. But this will create a 9:3:3:1 ratio. And these ratios you need to memorize. This is highly testable, super important. Okay, number 13 is what is genetic recombination? So for this one, I took the definition from the book, and the book says this is the process in which chromosomes are broken down and then rejoined to form new genetic combinations, and the process in which alleles are sorted and passed to offspring in combinations that are different from the parent. So this is basically saying, hey, look, your mom or your dad, they go through meiosis, they produce unique gametes, those gametes come together, and you create a unique individual. That's all a genetic recombination is. I'm just going to say mom plus dad equals you, and you are unique. You are the recombination of both of these genes. Okay, for question 14 is, what is the difference between a monohybrid cross and a dihybrid cross? So we kind of already talked about that, but I'll put it in words. Monohybrid is dealing with one set of alleles. So that would be like big P, big P. And then dihybrid is dealing with two sets of alleles, or two traits, which would be like big P, but big Y, and big Y. So this is one set, this is another set. Two sets of your different alleles, and then one set of alleles, your monohybrid. Okay, question 15, draw a dihybrid cross between TD, this parent type. So let's draw a big square. So to do this the long way, we're going to do a foiling technique. So big T is going to go with big D. Big T goes with big D. And then we're going to do it again. Big T goes with little d. Big T goes with little d. Then little t goes with big D. And then little t goes with little d. Oops. Right, so that's how you foil it. Now, I'm not going to do the second one. That's going to be the exact same. So let's just copy what we did up here on this side. All right, so let's make our grid. All right, so then now we're just going to do like what we do for a normal Punnett square. We're just going to put them together. So this one has big T, big D, big D. This one has big T, big D, little d. This one has big T, little t, big D, big D. Big T, little t, big D, little d. And we're just going to do that for all of these. Just keep going. It's a little tedious, but I'll show you ways to make this faster in worksheet number 4. But this is super important because it allows you to understand the ratios. Oops. So again, we said our ratio was 9, 3, 3, 1. So this 9 is going to be a dominant trait in all of the alleles. So let's highlight this. So dominant in all. So look at this first square up here. Big T, and big D. That's dominant in all of those. So we'll highlight that. And now look at the second one. Big T, big D, little d. Even though it's heterozygous for D, it still expresses all dominant traits. This one right here, big T, big D, still dominant in all traits. Big T, big D, little d, right? They're all heterozygous, but they're always going to express the dominant trait. So we're going to cut that green. And then that'll be the same for this one right here, this one right here, this one right here, this one right here, and this one right here. All these green ones are dominant in every single trait. It's dominant in T and it's dominant in D. If we count these up, right, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So that's where a 9 come from. 9 is dominant in all traits. Now our 3s are going to be dominant in one trait, but recessive in the other. So do red for this one. So let's look for ones that are dominant for T, but not D. So the first one is right here. This one's dominant for T, but not D, right? And then we go over here. We see this one. This is dominant in T, but not D. And then we need one more, which is over here. This is dominant in T, but not D. So that's where our first 3 comes in. Right? Now the other 3 is just going to be the opposite for that. Right? That's going to be dominant for T, or dominant for D, but not T. So this one's dominant in D, but T is not dominant. So this is 1/3. This one's dominant in D, but not T. So that's another one. Dominant in D and not T. This one, and then we're left with 1, which is our recessive for all. So your first one is dominant, your 9 is dominant in all traits. This 3 is dominant in one, but not the other. This one's dominant in the other, not the one that is in this previous 3. And then your 1 is recessive for both. Hopefully that should make sense. And this is exactly where Mendel got his 9:3:1 ratio, is from this dihybrid cross. Okay, so number 16 is, what does it mean by a person is unaffected? So this is something that was mentioned highly in class. So this unaffected thing is dealing with pedigrees, which is your fancy little family tree that has circles and squares, and it's typically looking at the transmission of a disease. So it's unaffected person. So just a little bit overview of a pedigree real quick. Circles are female. Squares are male. If you're diseased, you are shaded in. If you are unaffected, you are half shaded in. And this half shaded in box or circle, unaffected really just means heterozygous. Or the other term that they use is a carrier. So that's what you should look for. If it says unaffected, it means heterozygous. If it doesn't say unaffected, then it'll be called a carrier as opposed to affected, because it's carrying the gene. The box is half filled, but it's not affected. So this is the unaffected. Okay, so that should be it for all the lecture. Now we should move on to some practice questions. So the first practice question is, what is gametogenesis? The correct answer is D, both B and C, right? It's generation of sperm, spermatogenesis, generation of eggs, oogenesis. It is not zygote, because zygote is when you fertilize sperm and egg, right? That's fertilization, as after fertilization. So gametogenesis, generation of gametes, that's generation of sperm and egg. All right, number 2. When does the egg fully go through meiosis? Correct answer is A, fertilization, right? We talked about this when we talked about oogenesis. It's going to be arrested at meiosis. And then when ovulation happens, hopefully it'll meet a sperm. And then it will continue through meiosis. And then you'll form your diploid unique individual. It's not going to be at puberty. That's obviously wrong. While in the womb, not quite. And then at fertilization is the correct answer. Okay, so for number 3 is, where is the egg normally fertilized? Key here is normally. Normally it's the fallopian tube, right? The egg is actually released into the abdominal cavity. And hopefully it makes its way to the fallopian tube. Sometimes it doesn't. And that would be considered an ectopic pregnancy if that egg got fertilized outside of the fallopian tube, which is pretty harmful to the carrier, the mother. But normally, eggs are fertilized in the fallopian tube. Okay. Number 4. What is the correct association between genotype and phenotype? Right, so genotype is the code, so it can't be A, and it can't be D. And then phenotype is the expression of that code. So the correct answer is in fact C. Genotype is the code, while phenotype is the physical expression. B is wrong because phenotype is not the actual genetic material. That is wrong. C is the correct answer. Okay, moving on. What? Where did my paper go? So for question number 5, it's asking, what is the chromosome theory of inheritance? The inheritance pattern of traits that can be explained by transmission chromosomes. So B is the correct answer. A is wrong because chromosomes are being passed only by the mother. That's false. And then C, the pattern of chromosomes is universal. No. Alright, question 6. What are the genotypic ratios and the phenotypic ratios for Mendel's pea plants in experiment for a monohybrid cross between 2 heterozygous alleles? So the key word here is monohybrid. So wait, why do we see a 9331? That is a dihybrid. So we automatically know that A and C can't be it, right? That's a dihybrid. 9331 is a dihybrid cross. Now, B and D, we just have to make the correct associations between genotype and phenotype. And as we saw earlier, up when we did question 12, we saw that genotypically we had a 1-to-1 ratio, and phenotypically we had a 3-to-1 ratio. So the correct answer is in fact D. Okay, so in a certain family pedigree, you observe that 6 of the 9 couples' offsprings display disease in the next generation. Some of the children of the affected individuals have the disease, but none of the 3 unaffected parents do. You conclude that disease is caused by a blank allele. So I'd like to draw this one out real quick. So we're going to say we have 9 offspring. So let's just 1, 2, 3, 4, 5, 6, 7, 8, 9. And it says 6 out of the 9 display a disease. So we'll do this in a different color. So this one's going to be diseased. This one's being diseased. This one, this one, this one. Okay. So all the blue are diseased. Now, the next key information is in the next generation. So these kids here are going to have some other kids. Some of the children of the affected, so the blue, individuals will have the disease. So let's just draw an example. Saying if this one has kids, one might be unaffected and one may be affected. And then our next key information, it says, but none of the children of the 3 unaffected parents do. So that's saying that these blank ones are going to have unaffected children. So now we need to determine what type of allele this disease is, right? So our disease is the blue one. So if this, we're just gonna kind of guess around. We're just gonna try to observe things and kind of mentally picture what's going on here. So let's look at this guy right here. If this trait was recessive, how would it be possible that one is unaffected and the other is affected? That wouldn't make any sense, right? Now, let's say this disease is heterozygous, or this person's heterozygous. Then it would make sense that there's someone who's affected and someone who's unaffected. So we know at least the people that are affected, they have to be heterozygous, because if they're only recessive, then they're always going to produce the same offspring. And if it's always dominant, then again, we'll also get the same offspring. This is our true breeding. So we know that it can't be any of these. We'll have to assume it is heterozygous because we have a different progeny. One is affected, one is not affected. But to determine if the disease is caused by the dominant allele or the recessive allele, we have to look at the unaffected. So if we go back over here to this guy, we see that his offspring will not be affected whatsoever. So this can't be heterozygous because that would create a 1-to-1 ratio genotypically. And we see that everyone here, this is our true breeding, right? Regardless of how many self-fertilizations, it's always going to produce the same outcome. So we have to assume that this one's recessive, right? And if we're looking at the disease, right, it's likely going to be dominant because recessive would skip generations. And it would make sense for the unaffected, for this to be recessive and for the disease to be a dominant one. So B is the correct answer. Hopefully that made a little bit of sense. For these pedigree type things, you really just need to do puzzle solving. So just switch out alleles and see if it makes sense, right? When we put the recessive allele on the affected, it didn't make any sense, right? And if we did dominant, it also wouldn't make really sense because we're having an unaffected and an affected, and that's not expressive of traits that are homozygous, dominant, or recessive. So kind of just plug things around, kind of visualize, see if it makes sense. Draw your tree, that always helps too. So yeah, and we'll look more at pedigrees for worksheet number 4. This is just kind of an introduction question. So for question number 8, you don't know if a pea plant has green or yellow seeds. So to find out, you cross the plant with green seeds. This results in a mixture of yellow and green seeds. What would be the genotype of the, this is a typo here. It's supposed to be unknown. Pea plant. So they say, or we're going to say green is recessive, and then yellow is dominant. So this is a true breeding question. So we have an unknown, and to figure out, we're going to breed it with a plant of green seeds, right? That's our recessive. That is our true breeding. So let's just put this here, little y. This will be our unknown up here. But it says the result is a mixture of yellow and green seeds. So that's our huge hint. So just like we do with the pedigree, we're going to kind of plug and see if it makes sense. So let's say we think the unknown is going to be dominant. Well, if we do our little pedigree thing, we say that we get all recessive, or all heterozygous. Well, that would all express yellow. And we said before that is a mixture of yellow and green seeds. So it can't be homozygous dominant. So let's cross this one out. Let's undo, oops. Let's undo this little cross. So this can't be homozygous dominant, right? Because we say we have yellow and green. Dominant gives us all yellow. So let's try, what if it's recessive, right? Oh. Little y. That gives us all little y's. That gives us all green seeds. But we say we have a mixture of yellow and green. So that doesn't make any sense either. So then our last one, we're going to try our heterozygous. Just big Y, little y. Oops. Which gives us 2 green and 2 yellow. Which is exactly what happens here. 1 yellow, or yellow and green seeds. So the unknown must be heterozygous. That's the correct answer for 8. So just plug things in, see how they work. Just do a little Punnett square. It never hurts. Okay, so then number 9 says, recall that in pea plants, purple flower color is dominant while white is recessive. If 2 heterozygous plants are crossed, what is the probability that the first 6 offspring will have purple flowers? So this question right here is a product rule question. So let's look at this. Purple is dominant. So we'll do big P equals purple. Or I guess we'll just say P in general because it could be heterozygous. And then white is recessive. So little p is white. Now, if 2 heterozygous plants are crossed, so big P crossed with big P little p, what is the probability that the first 6 offspring have purple flowers? So for product rule, this works when there are independent events. And in this case, having different offspring are its own independent event. So let's do a little Punnett square. Big P, little p, big P, little p. So we get big P, big P, big P, big P, little p, big P, little p, and little p, little p. So we see it's asking is what is the probability the first 6 offspring will have purple flowers? So we'll do this right here. We'll do offspring number 1. What is the probability that it'll be purple? Well, we see right here in this Punnett square, 3 out of the 4, there's a 3/4 chance that it'll express a purple color. So for offspring number 1, it'll be 3/4. Now, because it's an independent event, we have offspring number 2, and it's also going to have a 3/4 chance of being purple, right? As we see in the Punnett square. And then our 3rd offspring is going to have the same exact chance. So 1, 2, 3, 4 offspring, 5, and then our 6th offspring. Right? And product rule, we're just going to multiply all of these across. So it's going to be 3 times 3 times 3 times 3 times 3, or just 3 to the 6th power, which let me plug this in the calculator real quick. Three to the 6th power is 929. And then if we do 4 to the 6th power, 4 times 4 times 4 times 4, etc., we get 4,096, which in decimal forms is 0.1779. Now we're looking for the probability. A lot of times probability is in percentage. Look at our answer choices. So to turn this decimal into percentage, we're going to multiply by 100. That'll give us 17.7%, which is closest to answer choice C, 18%. So this is an application of the product rule, and the product rule is only applicable if there are independent events. If the events are not independent of each other, then you have to use things like the binomial expansion equation. And we'll look at more examples like number 9 on next worksheet number 4. Okay, so for number 10, if the gametes of an organism are the same morphologically, they are said to be, keyword here is same. Correct answer is D, isogamous. So that's it for worksheet number 3. Sorry, got cut off at question 5 over here. But yeah, I hope you learned something. Continue working hard, guys. I will see you in the next worksheet.